Well, I'm going to talk about for 20 minutes or so about issues that I hope will be a springboard for some of the later, the panel discussion that's going to be led by Drs. Eastwood, Youngst, and Marshall, who I'll introduce after I give my talk here. And the object of my presentation right now is to kind of bring together some important concepts which I think have a lot of social and ethical implications for our country as well as developing countries and the relationship between those two entities. And so the first part of my talk, I'm going to really talk about what malaria is, brief overview on the pathogenesis of malaria and the significance of this health problem in the world today. Then I'm going to give you a brief background of how genetics studies before the modern genomic era. Of course, the modern genomic era 20 years from now will be quite different than the genomic era is right now. We always have a sense that we've completed something. In reality, it's obviously a continual process. But the kind of painstaking way that one has to do research to find out pathways or genetic, the underlying genetic basis of disease susceptibility and resistance to disease. And I'll just use that as a very simple example that came from our group that a postdoctoral fellow at the time, who's now at the University of Connecticut, Cheryl Patel, did on work she did in Papua New Guinea. I'm then going to talk about briefly about the hapmap, and both Patty and Eric have really been involved in this, and what the hapmap really is, and how this might provide some ways for us to kind of get better understanding of other genes or genetic pathways that may be involved in the pathogenesis of malaria, and the implication of this information and how it's gotten, what the implications are commercially in developing countries as well as developed countries, and for other disease processes that are not that important right now in developing countries. I hope I can pull that off. Okay, so first of all, what is malaria? Okay, this is, actually I started getting involved in malaria research actually in the middle part of my career. I was primarily involved in worm infections previous to that. And being on NIH study sections and looking and talking to several people who I got to know in that venue who were very much involved in malaria research, much more so than I was at that time, I was first of all impressed by the evolutionary biology of the organisms. The second thing that was fascinating, up until really the mid-80s or mid-90s, a lot of the most fundamental research on malaria biology, pathogenesis, and infection used mouse models. We don't have time to talk about this, but they ain't the best thing. You really have to go to the field to understand the pathogenesis of most human malaria, and that has implications for how you design a vaccine, how you prevent it, et cetera. So I saw this as a wonderful opportunity as a physician scientist to get involved in both basic mechanisms as well as in population-based studies overseas. And I think, so really the hapmap and other more recent genomic advances, both in technology and conceptually, has made this a fairly fruitful field for many research scientists here as well as in developing countries. So to briefly go over the way that the parasite is passed, there's an obligatory Anopheline mosquito that transmits the parasite. So when the female Anopheline mosquitoes takes her blood meal, now the driving force evolutionary for these Anopheline mosquitoes to take their blood meal is they absolutely need this protein in the blood meal to deposit their eggs, so it's really fulfilling the reproductive potential of the mosquito. So when the mosquito bites the person, they secrete saliva, which contains proteases, which actually are anticoagulants. So the mosquito can take up her 1 to 1.2 microliters of blood that's necessary to undergo oviposition. So a single-stage parasite called a sporozoite, within 15 to 30 minutes, must make it and successfully invade an hepatocyte. If the sporozoite, that stage, does not make it to the liver, within 15 to 30 minutes, it's absolutely gone. Actually, it's probably within seconds that it has to actually penetrate specific cells in the liver. Otherwise, it's destroyed in the bloodstream and by the proteases and other proteins in our blood. Within the liver, over a period of 1 to 2 weeks, in the case of Plasmodium falciparum, which is the most severe form of malaria we're concerned with, the parasite undergoes asexual reproduction. So a single sporozoite, by a single mosquito bite, then multiplies to form 20 to 30,000 forms in a single hepatocyte. Now, malaria doesn't directly cause liver disease. So even if your liver contains 10,000 hepatocytes infected with malaria parasites, it does nothing in terms of directly inducing liver disease. But after this week or two of asexual reproduction, these multiple forms, referred to then as merozoites with an hepatic schizance, are released from the liver cell. They then must, within minutes, or actually seconds, invade a red blood cell by a very, very specific receptor-ligand interactions that we're just beginning to uncover. Within the red blood cell, they undergo another cycle of asexual reproduction. So a single merozoite released from a liver cell infects a red blood cell. Over 42 to 48 hours, it undergoes asexual reproduction, multiplies into about the 16 daughter copies. The red blood cell then ruptures, and the reinvasion process in the red cells begins. So within 3 to 4 weeks after inoculation of a single sporozoite in an untreated individual, you can get a biomass of malarial parasites of 5 to 10 grams of malarial parasites, okay? So it's this exponential growth that accounts to a large extent for the remarkable transmission efficiency of the parasite. And here's some ring forms of Plasmodium falciparum infecting a red blood cell. Now the way that here's a kid here who has cerebral malaria. So how does this cause disease, aside from the obvious anemia? Well, in Plasmodium falciparum infection, these infected red blood cells, proteins synthesized by the parasite are transported to the surface of the red blood cells to make these red blood cells more sticky than they would be normally. These infected red blood cells with their so-called knobs on them actually then adhere to endothelial cells in critical organs, such as the brain, the lung. They have a propensity for being sequestered in the placental circulation. So this accounts for the major disease manifestations of malaria. The sequestration, which is essentially sludging, and the rheological properties of the blood are such that you get decreased oxygenation to distal tissues, ergo the cerebral malaria, pulmonary symptoms, and retarded growth retardation of the developing fetus. Here's a picture. This is an adult Thai patient who actually died of malaria, and you can see the petechiae throughout the white matter of the brain here. And this is a cross-section in the capillaries of the brain, and these are all infected red blood cells which have essentially clogged up the capillaries in the brain. So how does genetics help us with this? Well, this is one of the great Prussian scientists of the 40s and the 1950s, J.B.S. Haldane. Haldane, I think he was at Oxford, if I'm not mistaken, a population biologist. But he kind of put together, before there was convincing evidence that DNA encoded genetic information, is that he noted there was a geographic overlap of common erythroid variants and malaria endemicity in Africa. And the very classical example that we know of this is hemoglobin S. This actually happens to be, I think, the first protein that was ever sequenced, okay? So he, putting together theories of evolutionary biology at that time, he put together the concept that malaria, as well as other infectious diseases, are a powerful force of natural selection. And this, again, the driving force here is the increased reproductive fitness of our species. So hemoglobin S, to use as an example, is the example par excellence of a balanced polymorphism. So you have a single copy of it. Actually, if you do case control studies for severe malaria in Africa, you rarely, if ever, see any of the cases with hemoglobin S, okay, that are heterozygous for hemoglobin S, whereas if you go look at community controls, or kids who don't get cerebral malaria, who don't get severe malaria, the frequency of where I work in, for example, in Western Kenya, the mean frequency is 20% in the adult population. And I must say, in rural sub-Saharan Africa, I've never seen a homozygous individuals for hemoglobin S. That is sickle cell anemia. Why? It's because they're all dead. But they have, so it's the heterozygotes that are being promoted, that are being selected in the population. So with that kind of background, many groups have kind of looked at can we get any information on the pathogenesis of malaria and therefore rational preventative or treatment strategies based on looking at genetic variants in endemic populations? So one area that I work in as well is Papua New Guinea. And it's a very fascinating place. Linguistically, there's about 700 languages in Papua New Guinea. And there's a very interesting population biology in Papua New Guinea was probably settled, it's controversial, maybe 30 to 40,000 years ago by populations we would now refer to as Han Chinese. But it's, for those of you who've been there, it's a very rugged country. So founder populations are in one area, but there's different founder populations in another area. So it's a relatively small population. You not only get selective pressure, but you get population bottlenecks. So you go to one area, you see these very bizarre erythroid variants, for example, that are actually local to only that population in the world. So it's been known for years, based on blood group typing by serology, that the band 3, which is the most dominant protein in the red cell membrane, and glycophorin C, there are some specific abnormalities in red cells that are attributed to mutations in these particular proteins. And particularly glycophorin C is actually, we now know, encodes the so-called Gerbich blood group. And I would guess that everybody, I'd be willing to bet $1,000 that everyone in this room is Gerbich positive. Everybody in sub-Saharan Africa, whatever their background is, Hutu or whatever, are Gerbich positive. The only place in the world that you see Gerbich negativity is in certain areas of Papua New Guinea. Just by coincidence, we happen to work there. Actually, one of the first persons to study this was Carlton Gattacek, who got a Nobel Prize for the Kura work that he actually did in Papua New Guinea. But no one knew the molecular basis of this Gerbich negativity and what its significance it was. Well, to make a long story short, and I'll skip over this for the sake of time, the Gerbich blood group is a glycophorin C molecule. It's an integral red cell membrane protein, just like the band 3 protein is. And this is work done by Cheryl Patel. To make a long story short, we found that Gerbich negative individuals had ovalocytic red blood cells. Okay, so it led to, and this is in combination with another mutation in band 3, which is called, it's an anion exchanger. This results in a change in the red cell membrane fluidity. It also modifies the susceptibility of these red blood cells to infection by Plasmodium falciparum. So, again, that's kind of the traditional approach where you take a phenotype and you try to understand what the genetic basis of that phenotype is and determine how that relates to malaria susceptibility. And malaria and humans have been in this kind of evolutionary arms race. This is the so-called Red Queen hypothesis, okay? This is where there's a coevolution of two intensely competing species. It's analogous to an evolutionary arms race in that both species evolve continuously to gain advantage over their rival. Malaria, actually, its generation time is 2 weeks to 3 weeks. Ours is 21 years. But those parasites are also, through their evolutionary drive, having slight changes in their gene sequence in order to evade elimination in the mammalian host. So malaria, I think, is probably the best example of a human infectious disease that has exerted strong selection pressure on the human genome. There's multiple hemoglobin variants and red cell membrane variants. So how can we exploit this information to be useful for discovering new ways of preventing or interfering with the process of malaria, and how can we do this in the modern era of genome sciences? This is just, actually, I added this slide yesterday. And so the Red Queen hypothesis, which was Van Valen, I think, in 1973, a geneticist actually portrayed this, but it comes from from Through the Looking Glass by Lewis Carroll. So I read a bit about this yesterday. So the quote that I think is really pertinent to this, at the top of the hill, the Red Queen. Now, the Red Queen was one of the chess players that was Alice was chasing, okay? So the Red Queen begins to run faster and faster. Alice runs after the Red Queen, but is further perplexed to find that neither one seems to be moving. When they stop running, they're in exactly the same place. This is the issue, is that they're both moving, but they're both moving at a similar rate. It's kind of like the oscillations in the genome of a parasite and a host. You can see there's this oscillation here, which leads to this oscillation here, and over time, over generations, it appears that there's no change where they have come to kind of a terms with each other, so they have this kind of peaceful coexistence. Okay, so the hapmap and gene studies today. Okay, so an association study, put simply, is to identify genetic variants that are associated with the disease, so-called the cases, by comparing them for differences in the frequency of variants among a group of matched individuals with age, ethnicity, who do not have the disease, the controls. So this is the classical case control study, where if you do a study to say you look at 1,000 kids who are admitted to a hospital in Nigeria with cerebral malaria, you go out to the communities, you find kids who are age matched, who have no history of cerebral malaria, as best you can ascertain, and you compare them. If you did that today for hemoglobin S, you would find that difference in the frequency that I just had described a few minutes ago. So now we come up on these single nucleotide polymorphisms. These are single base changes that can be used as tags that are informative, at least statistically speaking, for nearby disease-causing or disease-preventing regions of the human genome. This will theoretically, and in reality, in the future, reveal previously unpredicted genetic regions of interest. So malaria susceptibility is undoubtedly polygenic. There's things that may prevent it, there are things that may lead to increased susceptibility, and there's to be negative and positive interactions. So this is really amazingly complex. There's a lot of simulation kind of work done on this. The other major concept that I think to keep in mind is the concept of linkage disequilibrium. This relates to the fact if you find a tag that's more common in the cases versus the controls, that tag doesn't necessarily mean that that identifies the specific gene or it's within the gene that causes the prevention or the increased susceptibility, but it may be related physically in proximity to areas of the genome of interest. And because of the size of the human genome, we're left with tags. We have the whole complete genome sequence. We don't know how those genes interact, and we actually don't know, we actually probably have a poor understanding of how these genes actually function in the organism as a whole. Okay, so what is the hapmap? And this was actually a slide that I took from Aravinda Chakravarti, who used to be at this institution, and he's now at Johns Hopkins, okay? So this is the international haplotype map of the human genome, okay? The goals are to define patterns of genetic variation across the human genome. I just said that. And the goal is to select single nucleotide polymorphisms. People use SNPs, but I hate the abbreviation, efficiently to tag common variants and then release all of these data to the public domain, okay? So phase 1 is completed, right? And I think phase 2 is completed too. 2.8 million markers in 270 people. So this is a remarkable thing. Of course, how much does this cost? We'll perhaps come to that a little bit later. So this is just the simple way that's constructed. You could gene sequence from different individuals. You find variants, and you want to find areas where these individuals actually all share a common tag so they can use those to more efficiently tag those areas in proximity to them. Okay, so the 270 people were selected as follows. There's the Yoruba population, which is an indigenous African ethnicity. There's the Ceph group, which are Caucasian people in Utah families. These are all trios, mothers, fathers, and offspring. Then there's the Han Chinese population from Beijing and unrelated Japanese individuals. And I just use this because this actually gives you, this is a remarkable thing in terms of its insight into evolution of the genome in different parts of the world. If you look, so the oldest population here is the Yoruba population. This is where all our African ancestors lived, okay? So over time, they have had the most time through recombination for this linkage disequilibrium to become shorter and shorter intervals simply through sexual reproduction. And you can see here in these areas of the genome here, for the Yoruba, this is the extent of various haplotypes. You can see that all these lines in general, especially like in here, are much shorter than in the Caucasian population or the Chinese and Japanese populations. So, who are the participants in this? It's an international group, Japanese, the Wellcome Trust, I've had these all listed here. Illumina is really now doing a lot. A lot of this is technologically driven to be able to do this in a cost-effective way, the Broad Institute, et cetera, et cetera. So it's an international consortium of all institutions from the developed world, okay, whatever the developed worlds mean, the industrialized world where the GDP is the greatest. So let's go to malaria for a moment. So we have not yet exploited the hapmap project for identifying gene pathways of disease prevention or susceptibility in human malaria. But we can use the information to see, well, if we didn't know about, these are a whole bunch of genetic variants that have been shown independently in the way that I just showed you for the Gerbich blood group to be thought to be involved in protection against severe malaria morbidity. So we actually, the hapmap was used, and two of these, this is hemoglobin S, this is a CD36, this is a molecule that's expressed in endothelial cells that's involved in cytoadherence of infected red blood cells, would have been picked out using the hapmap at this time as potential candidate areas. All these others here, which were found independently, were not found to be associated, would not have been picked out. Now, this doesn't mean the hapmap, some of these studies, for example, like this TLR4 and everything, are relatively small studies. They may not be sufficiently powered to do so. But this is kind of a glimpse to the future of how this kind of information might be used. So, before I introduce the next talk then, so I've been on several Foundation for NIH panels where we see 30 grants that come forward, and the deal is if you have the DNA in hand, and you show you have the intellectual wherewithal, and you have adequate DNA to do this, that's of good integrity, you apply to the Foundation to NIH, who then has given to them by either Illumina or Perlingen the chips to do these SNP maps with millions of markers on your samples. So I was on this panel a year ago, okay? So the diseases that were there, I was there because there was 3 on malaria morbidity, 3 on malaria, there were 6 on diabetes, there were 10 on Alzheimer's disease, okay? So we were the scientists, okay? So we had to kind of judge whether these are good questions, bad questions, and all these other kind of technical issues. So where is this so the real issue is that how will this information be used? And I think that really what's really coming up, particularly related to my work, are what are the ethical considerations of studying malaria in Africa in order to wittingly or unwittingly, which is more likely, advance knowledge and treatments of Western health problems. For example, we now know that ApoE and a certain allele of the ApoE gene is associated with a high risk of early onset Alzheimer's disease. Well, that genetic variant didn't evolve, or what isn't common in certain populations because it's known to some way lead to Alzheimer's disease at the age of 50 or 55. It could likely have evolved because of increased fitness to our species, because it helps decrease the severity of malaria, for example. So the question is that we make it information that could be used for actually Western diseases, which is all to the good, but how do you explain this to African populations, for example? And if there is profit in this, if Big Pharma takes this product and takes it forward, who shares in the profit for those kind of things? So I'll stop there. And I think it'd probably be better if I introduce our next speakers, and then maybe if there's any questions specifically what I asked, about what I talked about, I can address those. So we're going to have a panel discussion. Yeah, Dick, sorry.
Do you have a hapmap for malaria and comparing the malaria hapmap?
Yeah, right. So going back to this Lewis Carroll thing, yes, well, what we have for malaria, we have the complete genome sequence of malaria. But now a hapmap would involve taking different isolates from different parts of the world, and that is going forward, and it's SNP-based just exactly the same way. And that's really kind of one of the underpinnings of modern systems biology, if you will. I mean, this is a phenomenal amount of information that how do you integrate those two kind of approaches?